Pain falls under two common types, acute pain and chronic pain. Acute pain occurs after injury or surgery and is of short duration as it resolves once injured tissues heal. Chronic pain, on the other hand, is ongoing and lasts for longer than 6 months. For example, neuropathic and arthritic pain fall under this category. In chronic pain, pain signals remain active in the nervous system even after tissues have healed. A large number of individuals are looking for a solution for this issue. In fact, a rising 18.9% of individuals above the age of 18 have admitted to having chronic pain, with half of these cases persisting for the span of 10 or more years. With opioids such as fentanyl and morphine being the most commonly prescribed drug, opioid overdoses have increased to 115,000 fatalities worldwide in 2017, in part due to the increased use of opioids in the management of chronic pain. Opioids attach to proteins called opioid receptors on nerve cells in the brain, spinal cord, or other parts of the body. When this happens, pain signals from the body to be sent to the brain are blocked by the opioids. While they can effectively relieve pain, opioids carry risks and cons, such as barriers to accessibility and a variety of side effects. These include sedation, dizziness, nausea, constipation, respiratory depression, which in particular is very dangerous and often leads to overdose fatality, a cycle of drug dependence, and finally tolerance to the common opioids, which is defined by a reduced reaction to a drug following its repeated use. Increasing the drug's dosage may temporarily alleviate this problem. However, this may accelerate tolerance and further reduce the drug's effects. These individuals would be drawn to the streets for a cheap alternative to battle the increasingly fatal symptoms that accompany them. In due time, they would join the numbers contributing to the increasing death counts in North America. The opioid crisis, specifically in Canada, has been reported to have 10,337 opioid overdose deaths between January 2016 to September 2018. It is clear that a novel painkiller is needed, one that is safer than morphine and is efficacious. We have been looking into a synthetic version of pain relief in which we can modify and adapt our own body's pain-killing abilities. Within our bodies, our cells naturally produce a small peptide to regulate pain signals. This peptide is called enkephalin. It is related to endorphins with similar physiological effects of reducing pain. Its production is boosted in pleasurable activities such as eating, exercising, or even reading. However, when enkephalin is administered to patients from an external source, the peptide's path to pass through the blood-brain barrier is obstructed by the rapid degradation. Enkephalin is not stable nor available for uptake by cells, rendering it to be not as capable as opioids in its pain-killing effects. Hence, we introduced the KK103, which is an improved version of enkephalin. It is distinct by the addition of a chemical modification on one end of the molecule. This modification increases the stability of enkephalin, causing it to break down much slower, as well as increasing its availability to cells. Enkephalin, represented by the red line, declines very rapidly, while in comparison, KK103, represented by the blue line, shows promise in its long-term use as it is able to maintain its stability over a longer period of time. Further comparison through test tube study suggests that enkephalin is not able to permeate through the cell membrane, while KK103 passes through with ease. Thus, further animal studies show that the KK103 is longer lasting, has minimal withdrawal and tolerance, and lacks the symptoms and lethality of opioid use. Pain-blocking efficacy was tested between enkephalin, shown in green, morphine, shown in red, and finally KK103, shown in blue, with results showing that the KK103 maintains its stability better within 24 hours, while morphine spikes but declines again too rapidly. A series of experiments were conducted on mice who were injected with morphine and KK103, with the first experiment testing the drug's analgesic activity across 7 days. While the effect of morphine is shown to decrease, KK103 maintains its efficacy over the same amount of time. Another experiment compared the mice's dependence on the drugs by abruptly ceasing their long-term use and observing their reaction. As seen on the left side of the graph, the morphine group exhibits a much higher level of unease when the KK103 group showed none. Finally, breathing rates were measured to show that KK103 injected mice maintained their healthy breathing rate while respiratory depression occurred in morphine-injected mice. While favorable results have been produced from these experiments, research into this molecule is still a work in progress, with more overseeing to be done on the safety, metabolic, and efficacy profiles. Dr. Starley's research team at UBC are working together with the help of the Michael Smith Research Foundation and the Canadian Institute for Health Research.